So I'm going to tell you a story about the derivation of some of our work that's led to this, and then show you how we ended up asking ourselves the question of what, we've learned a lot about what PGRMC1 does. And I guess we were curious to learn, well, what regulates PGRMC1? One of the things, to cut to the chase, is that we've been spending some time interrogating certain microRNA and learned that LET7I may play an important role in regulating PGRMC1. And by inference, LET7I might influence the protective or death-promoting effects that we're talking about that seem to be mediated by PGRMC1, whether in isolation or in complex with a lot of different players that we're talking about. So just briefly, a couple of disclosures here, to say nothing of the fact that I'll be sharing with you about the potential use of estrogens and progestins in an off-label manner for certain diseases. So I wanted to, by way of introduction, share with you, I guess I'm supposed to stay here, that the average age of the menopause in the United States is about 51. And the mean lifespan for women in the United States, give or take, is about 81. And if you accept the premise that these hormones have very important effects on the brain, to say nothing of the other organ systems that it influences, the question that we asked is, what is the consequence of living a third of one's life in a hormone-deprived state? And whereas most of the history along the lines of this question is focused on estrogen and how estrogen may be neuroprotective or cytoprotective, we recognize that the menopause or the removal of the ovaries in both an experimental model or in oophorectomy represents a precipitous decline in not just estrogen levels, but estrogen and progesterone levels as well. I would like to just make a point that in the human menopause, whereas estrogen and progesterone levels drop quite significantly, androgen levels do not. So androgen levels kind of persist. And I may have time to talk a little bit about the significance of that as the talk concludes. But one of the things that we also started to appreciate, just as much as there's about 2- to 3-fold higher prevalence of Alzheimer's disease in women among men, there seems to be this shift also as a function of, maybe a function or coincident with the menopausal transition, where the risk for stroke changes. And this is some work, or actually a review, taken from a review published by Phyllis Wise and Dina Dubal, showing that whereas in this earlier age demographic, you see that men have a higher prevalence of stroke than women, but after the menopause, that shifts such that they've both increased, but there's less of a difference. And whereas the prevalence or incidence issue is somewhat debatable, what is very clear from the clinical literature is women have worse outcomes in response to stroke than men. So the question is why? And so in our thinking along the lines of what's the consequence of living a third of one's life in a hormone-deprived state, and whether or not that change or that reduction in those hormone levels could have made the brain vulnerable, we asked the question, is it possible that postmenopausal estrogen and or progestin therapy may be beneficial against certain age-associated dysfunctions or increases in the risks for certain diseases, to include stroke and Alzheimer's disease? So after making this sort of statement or hypothesis, we went down this path of asking, can estrogen be protective? What's the mechanisms by which it can be protective? And we found lots of literature and our own work to support the notion over the span of about a couple of decades. And this is one piece of work that we did a long time ago, where this was rats that had been overreactomized for 7 months. And what you see is the expression of BDNF. The warmer colors, the yellows and the reds, represent a higher level. The blues represent the cooler colors represent lower levels. And what you can see, there's nice abundance of BDNF in the cerebral cortex and in the hippocampus. but 7 months post-ovariectomy, there was a pretty profound reduction in BDNF levels. Okay, this is in situ hybridization, so we're looking at mRNA. And by the way, 7 months represents about a quarter of a rodent's life. So I want to kind of invoke again that what does it mean to spend a third of a woman's life in a hormone-deprived state? And what happened was, at least with estrogen replacement, and this was replaced 3 weeks after ovariectomy, so it wasn't an immediate replacement, we saw at least partial restoration of BDNF, at least in the hippocampus, but not the cerebral cortex. So one of the things that we inferred from this, or suggested, was that, well, maybe the incomplete restoration was a product of not having progesterone around. So we started to ask, well, could progesterone in and of itself have protective effects? And the short answer is yes. And this is a number of, these bullet points represent work from our lab and from many others that basically talked about how progesterone elicits effect, to say nothing of the fact that progesterone does in fact protect cortical neurons, and in our hands, in an ERK and PI3 kinase AKT dependent manner. So that was great. Progesterone is protective. But then came the Women's Health Initiative. And we were struck by that this very large clinical study seemed to yield results that were inconsistent with all this preclinical and some clinical data that had accumulated. And so we were curious why. And in fact, we came to the conclusion that there were at least 4 reasons that could explain the discrepancy. One was age, where the women enrolled in the memory study component of the WHI, they were 65 years and older. And I submit to you that it's not usual practice to initiate hormone therapy in a 65-year-old woman. And so that led us to ask the question, is there a finite window of opportunity? And I wanted to bring that up again because as we think about PGRMC1 or Sigma-2 targeted therapeutics, that maybe we should put our heads together to ask the question about, is there a window of opportunity by which these Sigma ligands or PGRMC1 ligands have protective effects? And the corollary, however, was that it may not just be biological aging, it could be the duration of ovarian hormone loss that could also be driving the change in responsiveness. But what underlines or underlies that? And so we started to ask which receptors are involved, and do those receptors change over the course of age? And so, with respect to progesterone, because we've been fairly hormone-centric in our study of the brain, we found that the classical progesterone receptor, that I abbreviate as PR, is essential for progesterone to elicit an increase in the abundance or cellular content of BDNF. So what you see here is the fold increase in, actually 6-fold, percent increase in wild-type mice that have the full complement of progesterone receptors in response to progesterone relative to the response to progesterone in animals that have their progesterone receptor knocked out. And you see that there was really no effect in the PR knockouts, but in the wild type there was, suggesting that at least the expression of BDNF, whether it's a function of stability, whether it's a function of de novo synthesis, that was critically dependent on the classical receptor. But clearly that wasn't the whole story, because we came to appreciate that the signaling effects that we had described previously, the activation of the MAP kinase pathway, the activation of the PI3 kinase pathway, they were not sensitive to the progesterone receptor antagonists, the pharmacological antagonists. So we said, well, is signaling and maybe another aspect of BDNF biology that we've been spending a lot of time on, that is BDNF release, how is that mediated? How is that effect of progesterone mediated? And so we started looking at PGRMC1. This is a progesterone receptor membrane component 1. And we thought it was a membrane-initiated event because we could recapitulate the signaling effects with these crude compounds that are progestins tethered to BSA. This bovine serum albumin conjugated progesterone that's connected by a carboxymethyloxine linker. Okay? And so here what you're just seeing is It'd be easier if I did this. This is just showing that we could knock down PGRMC1 with our siRNA approach. And then looking at BDNF release, so this is the amount of BDNF extruded into the media. Under mock transfected conditions with no progesterone added, this was our baseline. When we added progesterone, we got a nice increase in BDNF release. In the siRNA control, this was a scrambled sequence, nothing happens relative to the control. When we applied progesterone, when we just use a scrambled sequence, we still get that nice abundance of BDNF release in response to progesterone. But when we knock down PGRMC1, we completely lost the effect of progesterone, suggesting that the two receptors might really complement one another. It's the 1-2 punch. You need, on the one hand, the classical progesterone receptor to facilitate and build up the pool of endogenous BDNF, and then there's a separate mechanism that drives the release. But having both together is what drives it. And we've actually explored this mechanism further, and we've learned, quite surprisingly, that this effect on BDNF release is mediated by ERK5. ERK5 is also called BMK1, or big map kinase 1. And what I wanted to really show you is something that may influence our experimental design, because lots of us who might study the MAP kinase pathway might use a compound called UO126 or PD98059 to block the MAP kinase pathway. And we had published work with that as well, and lo and behold, both those compounds inhibit not just ERK1 and 2, but ERK5 as well. So we had to actually go back and ask the question, well, is it ERK1, 2? Is it ERK5? Which is it? And so what I want to show you here is when we knock down PGRMC1 with the siRNA, not only, we lost the effect of progesterone on ERK5 phosphorylation, but it had no impact on ERK1/2 phosphorylation, suggesting that the BDNF release that's elicited by progesterone is a product of ERK5. And to support this idea that you need this 1-2 punch, we looked at these astrocytes that have no classical progesterone receptor, but have PGRMC1. When we tried to protect these cells against iodoacetic acid, which is a combined a metabolic oxidative insult, you see reduction in cell viability in response to the insult alone. Progesterone at both 1 nanomolar and 10 nanomolar had no protective effects. But when we transfected the classical PR into it, now you have both the PR and PGRMC1, we were able to see a nice protection, suggesting that the two do cooperate. So Pixar animation, this is not, but just to give you a sense of what we think the working model is for these two. So progesterone can work through the classical receptor to drive BDNF production. But the release of BDNF requires ERK5 signaling, and that BDNF can then act on synaptogenesis, which is a subject of another paper that we published, and have neuroprotective effects, as has been already described. And so to some extent, this has also informed some of the pharmacology, because what we've also found is that whereas progesterone, the quote-unquote natural progestin, is protective, the one that's used in most hormone therapy regimens, MPA, or medroxyprogesterone acetate, is not. And what we think is going on is that it's actually very good at this, that is MPA, but it's ineffective here, or vice versa, because it doesn't allow for the 1-2 punch. In the short term, it's fine, but in the long term, it may have actually depleted. So what we wanted to ask, though, is, but what regulates PGRMC1? And so we turned our attention to some microRNA, and both PGRMC1 and BDNF, as it turns out, doing in silico analysis, have LET7I binding sites. So we asked the question, what would happen if we overexpress LET7I on progesterone-induced BDNF release? And short answer, when we apply the LET7I mimic relative to the control conditions, and here again you see progesterone eliciting BDNF release, when we applied LET7I, that progesterone effect was abolished. Interestingly, that was a product of LET7I and not LET7F. And I want to throw this out here, that the opposite was true when we looked at a model of Alzheimer's disease. So microRNA aren't universally effective. It depends on disease context. And that's another thing that we're learning. Work from Sung Cho in the lab, some of you have met him, have expanded the work from the astrocytes to also look at a model of neurons. And these are differentiated SHSY5Y cells. And what you find is that LET7I not only reduces PGRMC1 expression in the astrocytes, it also does so in this model of primary neurons. Excuse me, a model of neurons, which we've also extended to neurons, primaries. This is also true at the level of the protein. So this is mRNA and this is protein. So, okay, great. LET7I reduces it. But what happens if we kind of apply a age-associated disease, or age-associated pathology, I should say? So stroke. So this is a middle cerebral artery model of stroke. Middle cerebral artery occlusion model of stroke, I should say. And you see not only a reduction in PGRMC1, but you see an increase in LET7I to suggest that the two are related. And indeed, if we just increase oxidative load by applying hydrogen peroxide, you see a very similar sort of thing. You get an increase in LET7I, both in the astrocytes and in the model of neurons. So suggesting that this relationship exists, it's probably a product of disease, so much so that it might set the stage that prevents protective effects of a variety of ligands. Well, so then we ask the question the other way. What if we inhibited LET7I? Could we enhance the protective effects of certain compounds? And since we were focusing on progesterone, we asked the question in a stroke model. What you see here is a TTC stained is a product of stroke. So this is just the middle cerebral artery occlusion. You see a stroke in the cortical area. And this is just a rostral to caudal representation from the same animal. And what you see is the stroke causes a lesion. So where you see red, that's healthy tissue. Where you see white is ischemic tissue. Progesterone really didn't do much. In fact, it almost looked like it made it worse. Okay? But when we applied the LET7 inhibitor or antagomer, we got a very robust protective effect. This was seen at the level of motor function as well. And it basically what it did was it accelerated the recovery. So what you see here is the stroke, what happens in the stroked animal, which is this first bar, what happens with progesterone alone, and then what happens when you combine the antagomer to LET7I and progesterone. So you greatly accelerate the recovery. And over time, most groups sort of show a recovery. Progesterone is now better at day 7 versus day 3, but still you get this dramatic recovery. And now at 7 days, you've got complete restoration of motor function. And this was what really kind of captures it. And I should point out, this is 1 day after stroke. So what you'll see here is these are animals that have been stroked and treated with just progesterone, 1 day. And what you'll see is this stereotypical kind of circling motion because they have hemiparesis, and so they're kind of going to, they have one side of their body is able to move nicely and the other side is not. So they kind of circle. Again, 1 day after stroke, this is the animals that had been treated with progesterone and the antagomer. I couldn't tell the difference between the wild-type animals and these animals. And this is the subject of a patent that we've just recently submitted. So, but what I'd like to suggest is that cellular context matters. I think one of the things that we're very interested in is that neurons and glia interact. And that the expression of the right receptors are critical for progesterone to elicit its full-blown protective effects, and that what regulates some of those receptors include certain microRNA. I know there's a question. I'm just wrapping up now just to thank lots of people who help us do the work, to say nothing of the funding agencies that allow us to ask some of these questions. and the people along the way that have made critical contributions to the work. Particularly, I'd like to point out Chang Su and Natalia, who played a key role in driving the work that you've just seen, along with Trin Nguyen. So thank you. I'll be happy to take any questions you have.
Just real quickly, were the mice that were ischematized, were they mixed male and female, or were those only female?
No, those were just female.
Those are only
Yeah, yeah. So that's actually the phase 2, to compare this efficacy in males and females. Yeah.
Thank you. So I'm curious if you've compared isoforms of BDNF. And I guess what's motivating my question is, especially with some of the earlier slides you showed of in vivo BDNF differences, if some of them could be downstream of differences in neural activity.
Yeah, so we've tried to approach that one of two ways. We've looked at pro versus mature. That's one, and we see favoring of mature in response to the progesterone through PGRMC1. If we actually knock down PGRMC1, we lose that effect. The other is to actually look at in a 2-cell model where the hypothesis is progesterone works through astrocytes to release important growth factors to have protective effects on neurons. And that data would suggest that that's an important route. That's not to discount direct effects of progesterone on neurons, but a major effect, at least on the synaptogenesis side of things, appeared to be mediated by BDNF release from glia, then acting on receptors on the neurons to elicit increases in synaptogenic markers.
Okay, so great work. So I wanted to follow up on a comment you made about the I versus F being the opposite in Alzheimer's disease.
Good question. So that's actually the basis of a grant that goes out in February, because I'm not sure I'm smart enough to figure that out without having some funds to do the study, because in the 5X FAD mouse, it was LET7F that was increased, not LET7I. And yet, in our stroke models, it was LET7I that seemed to play the dominant role in terms of just the consequence of pathology. I can't speak to the intervention just yet, but the pathology seems to have a differential influence on the LET7 family. So is it possible that on the one hand, maybe amyloid is involved, or its processing is involved in a different way? Our very provocative idea that I want to just throw out there for critique is that perhaps any intervention that engages the BDNF system might benefit from inhibiting one or more of the LET7 family members. Because if BDNF is indeed so important for a variety of protective effects, whether it's effects on the cytoarchitecture of the cells, whether it's effects on long-term potentiation, if it's effect on intracellular signaling, differentiation, what have you, maybe we could augment some of the therapeutic effects of some new, or maybe even some of the ones that already exist, interventions, by enhancing the stage that involves the BDNF and its players.
Thank you. So one last question, if I could. So you mentioned earlier the androgen story. So maybe you could talk a little bit
Actually,
more.
I have to owe a debt of gratitude to Wayne, because one of the things that we've been unable to understand is, and I'll share with you the perspective. What I don't have time, because it's almost like a separate story, but it's parallel, is that we have identified an androgen receptor mechanism that it resides at the plasma membrane that seems to be sensitive to PGRMC1 inhibition. That is to say, but in this case, in the context of this membrane androgen receptor that we've been calling it, and actually membrane-associated androgen receptor, to distinguish it from Peter Thomas's MAR and other folks like AR45. In this case, when we activate through a PGRMC1 dependent mechanism, it actually favors cell death. Whereas here, in the neurons and the astrocytes, in a PGRMC1 dependent way, progesterone promotes survival. So based on some of the players that we're talking about, I'm really interested in having some follow-up conversations to help us understand why the difference. Because on the one hand, that death-promoting effect, we can mitigate by using siRNA to PGRMC1 or AG205. And I appreciate that there's some limitations of AG205. But these are also conversations I wanted to have with Bob, because I think some of the compounds you all have and some of the science that you've already built could teach us. So that's where that's coming from.
Thank you.